morning students. Today we are going to discuss 2 mark and 3 mark questions of plant tissue culture 5th lesson in 12th biobotany. Students, these questions are very important, and this is very easy topic. Plant tissue culture is very easy topic, and if you are studying all the questions, only lesser questions are here in this topic. If you are preparing for attending 2 mark, 3 mark or 1 mark or 5 mark, it will be very useful for you because you will get full mark. That much easy this topic. So please select this topic for answering 2 mark or 3 mark or 5 mark questions for your examination. First question is define explant. It is given in page number 97 under the topic 5.2 plant tissue culture. That is third paragraph given in green color box. Explant, the tissue taken from the selected plant transferred to a culture medium often to establish a new plant is called explant. Or you can write explant is the tissue of a plant to be cultured in tissue cultured is called explant. That also you can write. The next is define totipotency. It is given in basic concept of tissue culture in page number 97 in the second paragraph. Totipotency, the property of a plant cell that have genetic potential when cultured in nutrient medium to give rise to a complete individual plant. Or you can write the property of a plant cell when cultured in a nutrient medium to give rise to a complete individual plant is called totipotency. Then third question is define differentiation. Differentiation, the process of biochemical and structural changes by which cell becomes specialized in form and function is called differentiation. Then third question, define redifferentiation. It is given in page number 97, the third paragraph, third line. When the component of cells of callus have the ability to form a whole plant in nutrient medium, the phenomenon is called redifferentiation. The next is fifth question, dedifferentiation. It's also given in page number 97 below the heading redifferentiation. Open the bracket. The phenomenon of reversion of mature cell to meristematic state leading to the formation of callus is called dedifferentiation. Students, next question is define agar. It is given in page number 98. Agar is a solidifying agent used in plant tissue culture. That is, answer is page number 98. In the green color box, within the green color box it is given, agar is a complex mucilaginous polysaccharide obtained from marine alga used as solidifying agent in media preparation, tissue culture media preparation. The next is name some nutrient medium for plant tissue culture. Students, the answer for this question is given in page number 98 under the heading 5.2.2 technique involved in plant tissue culture. In the second heading media preparation, second paragraph, MS medium, B5 medium, white medium, niche medium, MS Murage Cook medium or Gambok medium or B5 medium, white medium and niche medium. Close the bracket. The next is, this is also important question. Define sterilization. How explant is sterilized? It is given in page number 98. Sterilization. First line itself. Sterilization is a technique employed to get rid of microbes such as bacteria, fungi in the culture medium, vessels and explant. Then second part of this question, how explant is sterilized? For this question, the answer is in page number 98. Explant sterilization. It is given sterilization of explant under the heading. The plant material also be used for tissue culture should be surface sterilized by exposing the material in running tap water and then treating it in surface sterilization agent like 0.1% mercury chloride, 7% ethanol under aseptic condition inside the laminar airflow chamber. The next is define callus. Page number 99. In page number 99, under the heading Induction of Calyx, fourth heading, Induction of Calyx, come to the last line, 3 lines. Calyx is a mass of unorganized growth of plant cell or tissue in vitro culture medium. The next question is define embryoids. Page number 99. Below that induction of calyx, another one heading embryogenesis is there. From that, the calyx cell undergo differentiation and produce somatic embryos known as embryoids. Students, next 11th question, name the different types of plant tissue culture. It is given in page number 99. Under the heading type of plant tissue culture, there are 4 types of tissue culture. They are organ culture, meristem culture, protoplast culture, cell suspension culture. Close the bracket. Students, next question is differentiate somoclonal variation and gametoclonal variation. It is given in page number 102. Somoclonal variation and gametoclonal variation is given in page number 102. Given in green color box, somoclonal variation found in plants regenerated in vitro variation found in leaf, stem, roots, tuber. Then gametoclonal variation or gametophytic variation found in plants regenerated in vitro gametic origin. Close the bracket. The next question is define artificial seed. Page number 102. Artificial seed. Under the topic 5.4.2 artificial seed in page number 102. Artificial seeds are synthetic seeds or produced by using embryoids obtained through in vitro culture. They may even derive from single cell or from any part of the plant that later divide to form cell mass containing dense cytoplasm, large nucleus, starch grains, proteins and oils. Close the bracket. Students, next question is name the material used for coating somatic seeds. Page number 103. It's given, answer is given in page number 103. The coating material for somatic embryo, first line, it is given in the first line. The coating material for somatic embryos are agarose and sodium alginate. Agarose and sodium alginate. The next question is define cryopreservation. Students, this is very important question. Cryopreservation. Page number 103. Cryopreservation is also known as cryoconservation. Is a process by which protoplast, cells, tissues or organelles, organs or extracellular matrix, enzymes or any other biological materials are subjected to preservation by cooling to very low temperature of, underline the number, minus 196 degrees centigrade using liquid nitrogen. Minus 196 degrees centigrade using liquid nitrogen. Close the bracket. Next question is 17th question. List out the cultural condition of plant tissue culture. Page number 99. Culture condition of plant tissue culture. Culture condition for plant tissue culture is given in page number 99 under the heading culture condition. The pH for the culture medium is 5.6 to 6. Temperature is 25 degree plus or minus 2 degrees centigrade. Humidity 50 to 60%. 16 hours illumination, photoperiods of illumination and 1000 lux fluorescence. Then aeration to the culture can be provided by automatic shaker or aeration of the medium by passing with filter sterilized air. The next question, close the bracket. The next question is 18th question. Define hardening. This is also a very important question. Hardening. It is given under the 6th heading, hardening, page number 99, second paragraph. Hardening is the gradual exposure of in vitro developed plantlets in humid chambers in diffused light for acclimatization so as to enable them to grow under normal field condition, close the bracket. Students, next is 5 mark question. So there are only limited question, 2 marks and 3 mark question, only 18 questions are there. If you are preparing this for answering in examination, then it will be very useful, because number of questions are very lesser, and this is very small lesson. Next one is 5 mark question. First question is, explain the basic concept of plant tissue culture. Under this concept of plant tissue culture, you studied 4 heading: totipotency, differentiation, redifferentiation, dedifferentiation. It is given in page number 97, and all the questions you already studied for 2 marks and 3 mark question, and that definition is enough for this basic concept. Before writing these 4 totipotency, differentiation, and what is redifferentiation, before writing this, first of all you write define tissue culture. Plant tissue culture. Plant tissue culture is culturing of plant tissue, cells or organ or leaf or any other parts of the plant in a controlled condition in artificial culture medium is called plant tissue culture. First you should write the definition for this plant tissue culture, and then only you write basic concept, 4 concept for the plant tissue culture, right? Then next is second question, list out the facilities available in a plant tissue culture laboratory. Page number 97 is given in page number 97 under the heading 5.2.1 laboratory facilities in plant tissue culture. First point is washing facility for glasswares and oven, drying glassware. The next is medium preparation, room with autoclave, electronic balance and pH meter. Transfer area, sterile room with a laminar airflow chamber. The next is culture facility, growing the explant inoculation in the culture tube to 22 to 28 degrees centigrade with illumination of 2400 lux photo period of 18 to 16 hours and relative humidity about 60%. Close the bracket. The next question is, what is sterilization and how culture room, culture vessels, nutrient media, explant are sterilized for plant tissue culture? Answer is given in page number 98. First is sterilization. The sterilization, defines sterilization also you already studied for 2 marks and 3 mark question. Then sterilization of the glasswares, forceps, scalpels, accessories in wet steam sterilized by autoclaving at 15 psi, that is 121 degrees centigrade for 15 to 30 minutes, and or dipping it in 70% ethanol by flaming and cooling. Then sterilization of culture room. For that, detergent, and then wash the floor with the detergent, floor and wall with the detergent, and then with 2% sodium hypochlorite and 95% ethanol. Laminar airflow is sterilized by 95% ethanol and UV radiation for 15 minutes. Likewise, sterilization of nutrient media. Nutrient media sterilization from culture media are dispensable from that point to 15 to 30 minutes. The next is sterilization of explant, that also already studied for 2 marks. So all the heading coming under sterilization must you should write. Then must write. Then next is list out the application of plant tissue culture. Page number 102. For this, answer is in page number 102. Application of plant tissue culture. There are 6 points given under the heading 5.4 application of plant tissue culture. Improved hybrid production, somatic embryoids can be encapsulated, production of disease resistant plant, production of stress resistant plants, herbicide tolerant heat resistant plant, micropropagation techniques of large number of plantlet for forestry, production of secondary metabolites from cell culture utilizing pharmaceutical, cosmetics and food industries. Close the bracket. The next question is define artificial seed. List out the advantages of artificial seed. Page number 103. Artificial seed. It is given artificial definition. For artificial seed definition, you have already studied this in artificial seed. Define page number 102. Artificial seed or synthetic seed, it is otherwise called synthetic seed, are produced by using embryoids obtained through in vitro culture. Then the advantages of artificial seeds is given in page number 103. There are 6 points. Millions of artificial seeds can be produced at low cost. Easy method to produce genetically engineered plants. Easy to test genotype of plant. Potentially stored for long time under cryopreservation. Artificial seeds produce identical plant. Artificial seed is greatly reduced. Dormancy of artificial seed is greatly reduced. Hence, growth is faster with a shortened life cycle. The next question is explain the techniques involved in plant tissue culture. For this, you should write, first of all, you should write define plant tissue culture. Then next, from the topic sterilization. Sterilization of the culture vessel, sterilization of the culture room, sterilization of nutrient media and explant. Then media preparation. What are the different media used for plant tissue culture? The next is culture condition, pH, temperature, humidity and aeration. Then how will you induce callus, induction of callus. How will you induce callus? And embryogenesis. How will you transform callus into embryoids or embryo? The next is after producing embryo or small plantlet, hardening of the plantlet. That is grow under normal field condition. Growing under normal field condition. How it is hardened. Up to this, you should write for this question, students. As I told already, this is very small lesson and limited questions are there. So if you are preparing this for your examination, you will get full mark for this. Thank you. All the best. Get ready for your examination. God bless you.